Science Unplugged, the podcast where science and conversation collide. Join us for a deeper look into the world of innovation. We are your hosts, Charvi Trivedi.
And the Slima Farooq. Let's explore the insights from the world of science. From the lab to the field, we'll dive into the stories and experiences of researchers, experts, and innovators who are shaping our understanding of the world.
Today we have with us Dr. Anupriya. She completed her PhD in biotechnology from Rajiv Gandhi Center for Biotechnology in Kerala and also has an MPhil in biomedical technology. She is currently working as a postdoctoral fellow at College of Health and Life Sciences, Hamad bin Khalifa University in Qatar. Welcome to the show, ma'am.
Thank you.
Let's start with the glowing stuff, literally. You work with something called genetically encoded bioluminescence resonance energy transfer or BRET based biosensors. That sounds like something out of a sci fi film. Can you break it down for us?
Thank you for asking the question, and it's a very interesting question. So, in order to understand properly about the breadth, I will break it down and tell you. I split each term and explain to you. And breadth is a very common phenomenon that is happening in the nature. You might have seen fireflies. So I will start explaining about the bioluminescence first. So the fireflies shows the light due to the bioluminescence reaction. So fireflies has an enzyme called luciferase, which oxidizes the substrate called luciferin, and as a byproduct, this light is being produced. So this phenomenon is exhibited by many other organisms, such as the marine organisms. I don't know whether you have seen the film Life of Pi. There is a scene where the whole water is glowing. That is bioluminescence. So when you come to the BRET, bioluminescence resonance energy transfer, that also is actually exhibited by many different organisms. For example, you might have seen jellyfish. Have you seen a color changing jellyfish? That color changing is due to, if you go to some aquarium, you can see color changing jellyfish, like from blue to green or blue to red. Likewise, you can see it when they move. So that is actually due to the bioluminescence resonance energy transfer. So I will break down the split that terms again. Resonance means when you play a musical instrument, when you click the string of guitar, that sound produces, that is resonance. So in more simpler terms, if you say, when you push a swing in a playground, that swing moves and that is resonating. So when you compare it with a trapeze so when an artist A and artist B is there, so they should have a proper wavelength for them to perform together. So once the artist A swings and come near to the artist B, then only, and their chemistry, their wavelength, their speed are similar, then they can perform together. This is similar to the PRET also. So when luminescent molecule, that is a luciferase enzyme, is in nearby a fluorescent molecule, suppose GFP, that is the common fluorescent molecule everybody knows. So green fluorescent protein. So if one luciferase enzyme and a fluorescent molecule are coming close together, and if their wavelength matches, like the emission wavelength of the luciferase matches with the uh, uh, excitation wavelength of the fluorescent protein, they shows BRET. The resonance energy is transferring from the luciferase enzyme, luciferase to the uh, fluorescent molecule, and you could see the fluorescence without any external light source. Usually the fluorescence, if you want to glow the fluorescence, you need to show the exact light, just as in confocal microscopy. Here you don't need. This is a chemical reaction. So if you add the substrate of the luciferase enzyme, that will produce an energy that is being transferred to the fluorescent molecule, which excites, and we are able to detect the proteins or the ligands associated with that in the cell without any external light source. So that is bioluminescence resonance energy transfer. So what we have done is we have developed biosensors. You know biosensors. Biosensors means glucometer. It is a biosensor. Any object that detects the biological molecule, that is biosensor. So in our case, what we have done, genetically encoded biosensors we have developed. So genetically encoded means this is a protein. We have generated a protein that is being coded by the DNA. Means we made a plasmid containing this particular biosensor, that is a protein, and so which has the luciferase molecule and the fluorescent molecule. So BRET-based biosensors we have developed. And in our study, we have developed the biosensors to detect the SARS-CoV-2 protein, so that this biosensor can be used in live cells as well as in vitro assays because they are protein. So that in live cells, we can monitor them in real time. So that is why we have developed a genetically encoded BRET-based biosensor. I hope you understand.
That was a very nice answer. What exactly is these biosensors used in for its applications?
Biosensor application. So in the one which we have developed, it can detect the SARS-CoV-2 proteins. So especially the protease of the SARS-CoV-2. So that if we wanted to identify a pharmacological inhibitor of the protein, we can use this biosensor. So in this case, in this biosensor, between the luciferase molecule and the fluorescent molecule, we have a linker sequence that link these two molecules using the SARS-CoV-2 protease cleavage site. So when the SARS-CoV-2 protein is there, this will cleave this biosensor so that luciferase molecule and the fluorescent molecule move apart. So this BRET, if the BRET to happen, there should be a that one particular distance they should be in. So this, when the distance increases, BRET will decrease. So the BRET means it is a ratio of fluorescence to luminescence. So when the this energy transfer won't happen, fluorescence values decreases. So as the BRET also decreases. So this is the way we detect how whether the SARS-CoV-2 protease is present or not. So luciferase molecule and the fluorescent molecule, we have a linker sequence that link these two molecules using the SARS-CoV-2 protease cleavage site compared to so when the SARS-CoV-2 protein is there, this will cleave this biosensor so that luciferase molecule and the fluorescent molecule move apart. So in this way, this we can understand if the BRET to happen, there should be pharmacological one particular distance they should be. So that is one. So of this when the distance increases, BRET will decrease. We can so the BRET means it is a ratio of fluorescence to luminescence. So when this energy transfer won't happen, fluorescence values decreases. So the BRET also decreases. So this is the way we detect how whether using the BRET SARS-CoV-2 protein is SARS-CoV-2 protease is present or not.
So we can say that biosensors are they can be a good tool for like diagnosing the presence of a particular virus more than the actual cure of it. Okay. So yeah, that was a very interesting concept that we actually talked about, because I had heard about FRET, but I had never heard about BRET. So yeah, that was quite
actually in FRET, two fluorescent molecules are there, and you need an external source of light. Here, one molecule is bioluminescence and the other molecule is fluorescence, and you don't need any external light source.
All right. Okay.